Hello, everyone. Today is the last Bio 240 lecture on immunology, and this is specific to the vertebrate immune system because we're interested in this unit in human biology. We talked a little bit about immune systems in general in all organisms, and we're going to come back to some structures and functions that are unique to vertebrates. So keep in mind that an immune system in general is going to help prevent infection from some other kind of organism or virus. Remember, there's no balance of nature, that every other kind of living thing out there, plus viruses, is trying to make its living. It's going to attack, take advantage of any possibility it has to gain its own needs, and if it causes harm to a host in the process, it doesn't care. That's its job. That's its ecological niche. All right, so when we think about disease, causes of disease or function or things that are going to cause harm to our human body, we have to think about it in the context of evolution, because as rapidly as our body is trying to fight against that organism or virus, that is going to cause a selective force on that organism or virus. And so if it has a mutation that allows it to evade our defenses or be able to survive and reproduce better in any way, then it's going to change in such a way that it makes it harder for our immune system to fight it, right? So our immune systems have been evolving throughout human history and are currently continuing to evolve even as we speak. It's one of the areas of the most rapid evolution in the human species. All right, so in general, the immune system has three jobs: to protect us from pathogens, and we'll have a good definition of what that is in the next slide. And also that we have damaged or malfunctioning cells in the body as well that are not because of a disease-causing organism. The immune system has a role in recognizing those and destroying them so they can be replaced by healthy cells. And in order to do these jobs, the immune system has a unique function compared to other body organ systems. It has to be able to detect what is yourself versus what is non-self. So this non-self recognition is critical for the immune system to work properly. And when it doesn't, we have goofy things happening like allergies and autoimmune disorders. So we'll be talking about some of the ways, the tools that the cells in your body have to know whether a particular cell or structure is supposed to be there. Is it part of your body? Should it be there? Is it self or a friendly organism? Or is it non-self, and does it need to be destroyed or removed? All right, so primarily we think about the immune system's job in helping us recover from disease. And so we're defining pathogens as disease-causing organisms plus viruses. Remember, viruses are not alive, so they are not technically biological organism. So viruses contain a small amount of DNA or RNA wrapped inside a protein capsule. In order to reproduce, they need to fasten on, they need to bind to a host cell. They'll insert that genetic material and use the host ribosomes in order to make the proteins that make up its genome. And then they'll destroy the cell as they are exploding out like the creatures from Alien, and have made new capsules in order to infect other cells. The bacteria, remember, those are prokaryotic organisms. They have differences in the molecules that make up their surface compared to eukaryotic cells. In the human immune system, we are a multi-celled eukaryotic organism. And so any disease-causing organism that's very different from us is going to be easier to detect. Whereas when we have fungal pathogens or single-celled or multi-celled eukaryotic organisms that can also be disease-causing that can also be disease-causing. Because they're eukaryotic, they're less different from our own body cells, so it gets harder and harder to detect and destroy different types of parasites. So based on their evolutionary history and their structure, their molecular makeup. All right, so disease-causing organisms can be viruses, bacteria, fungi, single-celled eukaryotes. So remember, there's all those things we used to call protists. Some of them are infectious diseases. The Plasmodium organism causes malaria. Trypanosomes cause sleeping sickness. So we have these single-celled organisms that can be pathogens. Usually we call the eukaryotic organisms, usually we call them parasites rather than pathogens. So there's multi-celled eukaryotes. Mostly these are worms, and so often they're called helminths like platyhelminthes. So like flukes or nematodes, so flatworms and roundworms. And we see these in veterinary settings as well. All right, so when we look at the study of these organisms, microbiology is typically the study of pathogens, and disease-causing organisms may also be called microbes, right? And principally bacteria are the things that microbiology is focused on. So if you're interested in diseases, microbiology would be a potential major. Now, don't get that mixed up with molecular biology, right? Microbiology is concerning microbes, right? And molecular biology is about molecules, which is going to principally focus on DNA and how DNA functions, how it does its job, and things that happen because of certain genes and other kinds of genetic information. So molecular biology is usually grouped together with cell biology, so it isn't necessarily focused on pathogens. It could be focused on development. It could be focused on other kinds of genetic characteristics that don't have anything to do with the immune system. All right, we're going to define an infection as a population of a pathogen that's sufficient to cause harm. So if we got one viral particle up our nose and it got into our bloodstream, that isn't going to make us sick. Our immune system's probably going to find it and kill it without us even knowing it's there. When we get sick, when we start to have a negative consequence or harm from that organism, it's because it has, at least one, has gotten into our body somehow, and it has started to reproduce. So think back to our population ecology topics, where we see that exponential growth of a population when it has a good environment that it's in. And so our initial exposure to any particular pathogen, that individual or few individuals is going to reproduce as fast as they possibly can. And then we're going to start to see illness because of the actual presence of that pathogen, or often because of the things our immune system is doing to try and kill the pathogen, right? So population ecology is really important in understanding how disease happens in the body, and then what the infection is trying to do, right? It's trying to increase its own fitness. It's trying to survive and reproduce. So it doesn't generally want to kill the host, because if it kills the host, then all of its individuals die. But it also wants a way to spread to another host. All right, so it's going to try and survive long enough in one host that it gets spread to another host. So really interesting aspect of the study of diseases is how they spread, how they get from one host to another. So there's different mechanisms. We know there's sexually transmitted diseases that happen through contact of the genitals We have blood-borne diseases where you have to have, they're carried in the blood, and you have blood that's moved from one individual from another. The most common types of infections are spread through fecal waste, right? So when we don't have a good water treatment system, we don't have good separation of sewage from drinking water, it's really easy to get digestive diseases spread from one individual to another. We don't have good sanitary practices in our culture. We can have fecal material spread from one individual to another through the preparation of food, after changing a baby diaper, or not cleaning, or cleaning oneself after making fecal matter. And then we have respiratory spread. So when you sneeze or you exhale strongly, and little particles of mucus come out of your nose and mouth, and those hang around in the air, and another person inhales them, or they fall onto a surface and somebody touches that and touches their face. All right, so the infection wants to get to a new host, right? So the host is the kind of individual that can be attacked by that particular pathogen. Most pathogens have very specific hosts. They may only be able to infect a single species, right? And that the way they do that is that each organism has certain proteins that are sticking up from the surface of its cells. The pathogen has to bind to those receptors. It has to be able to detect in some way that this is the right kind of cell to attack. It has to have a structure that is actually physically matching up with the surface structure of the host, right? So most pathogens are host specific. There may be cross reactivity, or an infection may be more general, right? So we're seeing, for example, that COVID, some other kinds of organisms can become infected with it. Zoo animals, some kinds of big cats and gorillas, for example. As pathogens evolve, they may have an event that happens, a mutation that allows them to switch from one host to another. So most human pathogens have evolved in some other kind of organism, and then had a genetic change that allows them to switch. So some kinds of host organisms might be primates because they're very similar to us. Other mammals, such as bats, possibly birds, have been places where certain diseases have come from, in terms of jumping species or switching what kind of organism that they can infect. Right. All right, so once we have an infection that's happening, when we have the pathogen inside the body, they may penetrate or go inside of host cells, or they may exist outside of the host cells. They may be circulating in our bloodstream. They may be too large to get inside of a cell. Right, so our immune system has to fight those different kinds of pathogens in different ways, based on how it can access them in order to try and destroy them. So when we actually get sick, how do we feel? We don't feel good. So some of those symptoms are just a result of how much energy that it takes for our immune system to mount a response. There's tons of cell division. There's tons of molecules that are being produced. It takes lots of energy. We feel tired. The pathogen may release a molecule as part of its natural metabolism, or as a way to attack the host specifically. So these molecules, we usually call them toxins. And so the damage that's happening to our body, or the symptoms of the disease, may be a result of this toxic material. Tetanus is a disease where a toxin is released into the body. Most often, damage to the body is done by viruses. It actually goes inside of a body cell, and then it destroys the cell. So we're having damage done to cells that are inside of the body by the disease. And an accumulation of a large amount of that damage can cause organ failure, or in the most severe cases. Other things that we experience as a result of being infected by a disease are actually processes that our own immune system does in order to fight the pathogen. So for example, inflammation, fever, diarrhea, these are all responses to the presence of the pathogen that the immune system is doing in order to try and fight it. If these symptoms that are caused by our very own immune systems go on too long or too extreme, or for an individual that isn't otherwise healthy, it's the immune response itself that can actually cause the harm to the body that can lead to serious health consequences or even death, right? We have a fever that's too high for too long. That can be fatal. All right, so a lot to process. We're going to sort of keep going back over some of these ideas and see how the immune system does these things as we go through this lecture. All right, so in vertebrates, right? So we're talking bony fishes on up. The vertebrate immune system has two components. Whereas in other organisms, at least as far as we know now, this may change in the future, that we have a sort of an extra bonus added component of our immune system and certain other types of cells and responses that are unique to vertebrates. So we have these two portions of the immune system: innate immunity, or sort of internal immunity, and then acquired immunity. Sometimes it's also called adaptive. All right, acquired immunity is added on to our innate immune system. So the innate immunity is a set of responses that are always on, they're always there, they're also sort of always going on at a certain level, and they're going to prevent infection. They're going to be non-specific. That means over human evolution, we have been attacked by a variety of viruses and other microorganisms. And we have a general idea of the kinds of organisms that are non-self, that are pathogens, that we need to be able to protect ourselves from. Right? So they're general. They are always on. And they're rapid in the sense that they're always there, they're always present, they're ready to go at a whenever you get infected by anything. All right, and in all organisms, like we talked about in plants, even in single-celled organisms, there's an intact barrier around the outside, whether that's a cell membrane or a more complex organ, the skin, where we have living surfaces, live cells, our eyes, the inside of our mouth, our digestive tract, the inside surfaces of other parts of the body that are in contact, the urogenital system. They're covered with mucus, right? Mucus isn't just sitting there. Mucous membranes are the layers of epithelial cells that are on these live cells. They're going to be secreting mucus actively. There are certain types of cells that live in those epithelial populations that also are able to do immune functions. And we're going to have a variety of molecules besides just mucus, but other molecules we talked about in the digestive tract. Lysozyme, that's an enzyme that's in our saliva that can break down certain types of pathogens, cell membranes. We also have slightly acidic solutions that are secreted by our barrier defenses. So low pH is going to help protect against pathogens in a couple ways. When we look inside the body of a multi-celled organism, the main type of immune cells we think about are these phagocytics. So we have large, typically amoeba-like cells that are going to phagocytose or do endocytosis around pathogens. So especially for bacteria, we've got bacteria floating around, these phagocytic cells are going to bind to them and engulf them and destroy them. Other types of cells may be secreting antimicrobial proteins, a variety of different molecules like lysozyme. In vertebrates, we have extra added bonus component parts of this rapid response, always on response. We have something called the inflammatory response. We'll talk about that more in a few minutes. And we have a type of cell that's circulating in our blood called natural killer cells, and they're capable of recognizing and destroying some types of pathogens. All right, the acquired immune system is really interesting. It is able to, by a variety of really cool mechanisms we'll talk about in a few minutes, recognize the particular pathogen that you're being attacked by right now. And it's going to increase the population of cells that are able to attack that specific pathogen. It takes it a little while to do this, right? So it's a slower response. It may take days to weeks in order to completely turn it on and bring it up to speed. And the other component of it that isn't shown on this slide is that a portion of those cells are not going to be used to fight the current infection. They're going to stay in the circulating bloodstream, waiting until that infection attacks you again. And in that case, they're going to remember it. And so we also have, as part of acquired immunity, not just that it's going to recognize a specific pathogen, but it's going to create an immune system memory of that pathogen so that you will be able to fight it off much better in the future. Acquired immune system is focused on certain types of cells. There's B cells and T cells, and they're going to do things like release antibodies into your circulating bloodstream. And also this cell-mediated response is that certain types of cells are actually going to attack the infected cells in the body. So not the pathogen, but the host cells, your own body cells. All right. So thinking about the immune system again in a slightly different way, it has lots of different component parts that interact in different ways. So I have a couple different sort of outline slides in this lecture that are thinking about the immune system in different ways. So one thing we do in our organismal biology is we talk about structures and functions. All right, so we have a couple different parts of the immune system function. So just to organize the things that a good immune system should do, right? We have a really good one. It has to be able to identify the pathogens. It has to be able to respond by killing the current infection. And remember that everything in the body takes energy. And that if we're going to have an immune system that is going to fight a disease, we don't want to be wasting a ton of energy all the time in order to have some amazing amount of immunity that is constantly on. So we want to be able to turn the system on and have it ramp up or be upregulated, turned on and up, make it get louder, right? Amplification. But only when necessary. Right? We don't want it to respond. We don't want to turn it on when we don't have a pathogen to deal with. And then once the disease-causing organism has been, for the most part, eliminated from the body, we need to turn it off, right? So it needs to have an on switch and an off switch. That's called retreat. And ideally, we want to have it remember once we want it to learn, right? We want to be able to prevent future infections. So when we think about the different parts of the immune system that we've talked about already, the innate parts are able to do the pathogen identification, and they don't do a specific response, they do a general response to the current infection. So we have a difference in function between the innate parts of the immune system versus the acquired. So the acquired immune system, those component parts are mostly responsible for the specific response, the amplification, retreat, and are 100% responsible for the remembrance. we think about what parts of the body participate in the immune system. Lots of them do. So when we think about our major body systems, we have the integumentary system, which mostly consists of the skin. That's an organ system that is going to prevent infection. So we can think about it as being part of the immune system. The main body organ system that we think of as having a specialized job of immune functions is called the lymph system. We'll look at it in more detail in a few minutes. Most of the immune system cells that are circulating in the body are part of the blood. So the circulatory system consists of the pump, the vessels, and the body fluid. So the blood is part of the circulatory system. So we consider that a part of the immune system. And then we have some component parts of the digestive system that have immune functions. There's little patches of cells. There's structures associated with the digestive system. So we can think of that as a system that doesn't have a primary job of doing immune system functions, but it participates in immune system functions. Right, so skin and lymph are the biggest ones, circulatory, because that's where most of the cells live. Right, so we think about organ systems, right? We're going to look at specific organs in a few minutes, but when we come down to cells, most of the work of the immune system is done at the cell level. Right? The pathogen is made of cells or virus, and so the attack, or how do we destroy the pathogen, is done at a cell-to-cell level. So there are both white blood cells and what we would call white cells or immune system cells that live embedded in the surfaces of tissues, right? So embedded in tissues or at the surface of the epithelium. I'm not going to focus in this lecture on a sort of a set of types of cells and what they do that are part of this epithelial system, but they function for the most part in more or less the same ways as what's happening with the white blood cells. So we do have these sort of two components, circulating in the bloodstream and also embedded in tissues and or at surfaces. So we have, like, say, in the surface of the lining of the digestive tract, the lining of respiratory surfaces, there are embedded immune system cells. We don't want to call them blood cells because they're not in the blood, they're in tissues, but they have similar functions to ones that are circulating in the bloodstream. And we'll see that some of the cells are going to migrate back and forth to different places where they're needed. There's a variety of molecules that are really important in the immune system, right? Starting with the receptor proteins that are sticking up on the surface of the cells, right? And then how the virus or the bacteria binds to your own body cells, the kinds of receptors that are used in the immune system itself so that those cells can recognize one another. The immune system cells may be secreting or releasing proteins that have an antimicrobial function in some way. We'll look at some examples, not just proteins, but other molecules as well. All kinds of crazy things. Antibodies are a protein that is released by certain type of B cells, plasma B cells. Antibodies are often what we talk about in terms of what we get from a vaccine. And the reason why they're important is that they have a job of actually trying to help the body get rid of that pathogen. And we'll see how they do that job in a few minutes. And then cytokines. So when we think about communication between different parts of the body, different organs, different functions, we've talked about hormone molecules before. In organismal biology, the immune system has its whole own set of communication molecules. And generally they're called cytokines. There's dozens of them, and they work, they have different functions in terms of what kinds of messages they send to one another. And we'll come back to that in a few minutes. So when we look at, excuse me, as we're thinking about how the body can prevent, especially prevent pathogen infection, think about how we try and prevent food from spoiling, right? So food spoils two things can happen. It can become unpalatable, that is gross, and we don't want to eat it. But it can also become a source of disease, right? We can get botulism, poisoning from certain types of spoiled foods. It might give us, there's a bacteria that can grow on them that can make us have a, can be very serious digestive disorder. Right, so how do we prevent that? Well, if we refrigerate or freeze our foods, what that does is it doesn't necessarily kill the organism, but it slows down its metabolism so that it can't reproduce, right? We've got that potato salad sitting out on the picnic table, and it's got some bacteria in it. We can't prevent them from existing everywhere, right? But what happens is they start to divide and they start to grow, increase their population while it's sitting out on the table in the afternoon. If we keep the potato salad in the refrigerator, it's too cold for them to reproduce, right? So we prevent them from growing enough bacteria in that bowl of potato salad that when we eat it later for our midnight snack, that we aren't putting so many bacteria into our gut that they're able to overwhelm our immune. So little tiny dose bacteria, okay, we just get rid of it. Larger dose of the bacteria, we will get sick. So what else can we do? Right, we don't think about it too much nowadays because refrigeration and freezing is so easy. But when we cook our food, we're also killing a lot of bacterial contaminants. It might be in there already. That also kills a variety of eukaryotic types of organisms, such as parasites. Freezing can kill eukaryotic pathogens. For example, if you're eating sushi, there can be some parasites that are in fresh raw fish. Usually nowadays it's frozen very rapidly, and that kills any pathogens that might be in there. It also makes it easy to slice really thin. When we can foods, right? So why can we keep a can of food on the shelf for literally years and it's still good when we open it up? It hasn't had pathogenic molecules or organisms invading it and growing. It's because before we put it in the can, we boil it or clean it, prepare it in some way that we think there aren't any pathogens that are going into the can in the first place. And then we remove most of the air that's in there, fill it up with liquid. And then when we seal it, there's almost no oxygen in that space. Oxygen may be required for a pathogen to survive, so we're cutting it off from that. Many of the canned foods that we make may have a really high amount of vinegar or sugar in them. That also helps prevent bacterial and viral growth. Even if we don't can or seal the foods that we're pickling, having that really low pH of pickles, a lot of vegetables that we pickle, those helps prevent pathogens and also fermentation. So products like sauerkraut or kimchi, those are going to have this process going on that can actually suppress pathogenic bacteria as well. The other thing we do with foods is dry them, right? So we have dried fruits, we have dried meats like jerky. Smoking as well is a way of sort of cooking the food so that it kills any pathogens that might be in it. And so if we remove all the water, right, living cells have to have a watery environment in order to survive. Right? So if we dry the food completely, then there's no way for those pathogens to survive and reproduce. Right? So where we have the surface of our skin, it's dry because it's these dead cells that aren't alive. That's going to prevent the growth of most pathogens on the surface of our skin. Where we have live mucous membranes, things like our eyes, we're constantly flushing the surface of our eyes with tears, right? So we're rinsing away any potential pathogens. When we look inside our respiratory system, it's not only covered with mucus, but right? So the mucus is a physical barrier. A lot of bacteria, viruses can stick in it. And then not only do they stick in it, but we have constant ciliary action. And so cilia on the surface of all of the epithelium, like a little elevator, it's constantly pushing the mucus up to the back of our throat, where we can either spit it out or swallow it. In our digestive tract, we will digest and break down those potential pathogens. Our other systems, the digestive tract, the urogenital tract, we're flushing out urine, we're flushing out material that's in our gut that might be potentially pathogenic. And those are all ways that we try and prevent infection in the first place. And if we do get a cut or a scrape on our skin, what does our body do? Heals it up as fast as it can, because that would be a way that infection can get into the body. Right, so I've talked about a few of these things already, but again, this is a way I want to organize the information for you. So what are all the kinds of things the immune system can do? And then a little bit more specific on what are the tools that we have to do it, right? So these are structures, right? Structures, what job do they do? We want to be able to prevent infection. That's again the intact skin surface, low pH, lysozyme, mucus. And then we have friendly bacteria, mutualistic bacteria that live in our gut, that live on the surface of our skin. We're going to use the principles of ecology again in this lecture, where we have a healthy population of friendly bacteria, ones that are not pathogens. They're going to compete with, they're going to competitively exclude, hopefully, they're going to prevent an infection from pathogenic bacteria because they're taking up that ecological space, right? Friendly bacteria. So these are ones that we might call probiotics or our gut microbiome. And so like we talked about in the digestion lecture, different mixtures of our gut flora, our friendly bacteria, can have a big effect, or at least we're starting to develop an understanding of what the community of bacteria on or inside our bodies can have, how it influences our health. All right, so some of the things our body does, our immune system does, is to try and reduce the ability of the pathogen to multiply. All right, so once we detect that there's a pathogen inside the body, it's probably a virus or a bacteria. What circumstances do the virus or bacteria need to have in order to increase in population? Well, a virus in particular needs to be able to synthesize proteins, right? It's going to attack your cell, insert its genetic material, and then make proteins. It can't increase in numbers unless the cells of your own body are actually making the proteins in order to make the viral capsids. All right, so one of the things that fever does is it decreases the rate of protein synthesis, and also some of the other factors that are released or molecules that are released are going to sort of downregulate neighbor cells from doing protein synthesis. So basically their own metabolism is going to pause, and it's going to wait until the infection is removed before they go back to normal. Fever, or the increase of body temperature, may actually also make it easier for your immune system cells to find and stick to pathogens, right? So it may have multiple functions that we're just starting to learn more about. In order for your body to kill or destroy in some way or inactivate pathogens, we have these phagocytic cells that are going to be able, there's several kinds, but they're actually going to grab onto and eat, basically. That's what phagocytosis is. But we also have another set of proteins called the complement system. Complement system is is fairly complicated, really interesting. It's a series or a group of different proteins that are circulating in the bloodstream that are always there. However, they're triggered to do a series of chemical reactions under certain circumstances. And one of the things that can result is that they form a thing called the attack complex. The attack complex is actually some of these proteins from circulating in the bloodstream. They're going to change their form and they're going to stick together, and they actually make a ring. And that ring is going, or it's going to insert itself into the cell membrane of the target cell, and it's going to open up, right? It's basically punching a hole in the cell membrane. And so what happens when when a single-celled organism has a big old hole in its cell membrane, is that all of a sudden its cell cytoplasm it's not going to be able to regulate its internal conditions anymore. And basically, if enough of these holes form in the cell membrane, it basically explodes and dies. All right, so this is pretty crazy. This is like science fiction stuff. So the other thing that we have to do is not just destroy sort of these pathogens that might be floating around in the circulatory system, but we have cells of our own body that are infected by the pathogens. So we have intracellular pathogens when we have gone inside, right? So if we have pathogens that are on the outside of cells, they're in spaces in the body. Those are extracellular, outside the cells. We have intracellular pathogens, right? They're hiding inside the cells of our own bodies. So what we have to do in order to fight those is actually destroy the cells of our own body that are infected by the pathogen. And the way that our body does this, it sort of has a two-step process. One is that there's some proteins that are called perforins, right? If we think about paper that has holes in it, it's perforated. So perforin is similar to the complement system in that it makes a hole in the cell membrane. Doesn't make quite as big a hole. And what happens is this little packet of perforins and this stuff called granzyme is secreted by the attacking cell. And the perforin makes a hole in the cell membrane, the granzyme gets in there. And what it does is it binds to the DNA of the body cell, and it turns on a series of genes that actually cause the cell to self-destruct, right? Apoptosis, if you think about a cell popping, it's dead. Apoptosis is the word for this programmed cell death. All the cells in our body have this, self-destruct button intrinsic. Important processes in development happen because certain structures grow, and then in a pattern, the cells are destroyed. So when your little hand is growing as an embryo, it actually grows with webbing in between all the fingers as it's starting to grow. And then the tissue that separates the fingers disintegrates by apoptosis. Those cells are triggered to die in order to separate the fingers from one another. All right, so this combination of perforin and granzymes is used to kill cells that are infected. Right? That prevents the infection from spreading. Kind of like in those movies when you have, there's a breach in the hull of the submarine and everybody's trying to run out, and there's the watertight doors. We're going to sacrifice some part. Some guy is going to get stuck behind the watertight door with his head on the base in the portal, and he's drowning, and everybody else on the ship gets saved because that compartment got separated off. Kind of like that. All right. And then we have these memory cells that are going to be able to help prevent future infections. All right. So now we're going to start talking about this lymph system. What is it? Right? What does it look like? It's a set of tubes or vessels that are distributed throughout the body, kind of parallel to the circulatory system, not quite as extensive. It is associated with the circulatory system. We've already seen that it's associated with the digestive system. And although it primarily has an immune system function, it also coordinates with the circulatory system. So it does transport fat molecules that are coming from the digestive system that are too big. We talked about those lipoproteins, the fats that are coming from the small intestine that go into the lymph system. The reason that works is because we have this region, these large veins that are near our neck and our heart, and the lymph system actually is connected. The lymph fluid flows into veins that are paired with them at this location in the body. The other thing the lymph system does in coordination with the circulatory system is anywhere that there's capillaries that are delivering nutrients and oxygen to the tissues. Remember that we have a little bit of plasma that's forced out of the capillary. Right now, a lot of it, most of it should get sucked back into the downstream capillary because of the gradient in pressure that's created. But some of it, and under certain circumstances, a little extra might remain. So that interstitial fluid that can accumulate in tissues, it's easier for it to get back into the lymph system than it is back into the circulatory system. So the lymph system is also helps collect extra interstitial fluid, right, and turn it back into plasma by dumping it into the circulatory system. The fluid that is inside the lymphatic vessels will be called lymph. Where the lymphatic vessels branch from one another, there are these larger structures called nodes. The nodes are kind of a little maze inside of them. They're packed full of immune system cells. And so basically what they do is they act like filters. They force the lymph fluid to go through this little narrow pathways that allows any pathogens that are in the lymph fluid to be grabbed by the immune system cells that are concentrated there. All right, so we have these lymph vessels, the lymph nodes, and then we have some accessory glands that are related to the digestive system, or structures, the adenoids, tonsils, appendix. These are structures that we think have some immune system. They may have some extra immune system cells associated with them. Now, you may know that you can get your tonsils or your appendix removed and you don't die from that. You can live without it. We are not 100% sure of what the functions of those organs are, but we think they have some immune system function. Inside the small intestine itself, we have these Peyer's patches. They're little concentrations of immune system cells, like I talked about before, where we have epithelia. We're going to have embedded in those cells or mixed in with those cells some immune system cells that are surface associated and that are going to be trying to screen and grab any pathogens that are in the digestive tract. The spleen is a major immune system structure, but what it does is it filters blood. It doesn't filter lymph. And so blood is flowing through it. Its structure is basically similar to the lymph nodes in that it has a concentration of immune system cells. The other thing the spleen is responsible for is removing old and damaged red blood cells as well. Let's see what else. So these nodes can be infected with or have cancer. So if you have a woman or a man, say, has breast cancer, it may have spread to nearby lymph nodes, right? So if the tumor is being removed, nearby lymph nodes may also be removed. And what happens sometimes is if these lymph nodes that are near the armpit have to be removed because they're cancerous, what it does is it cuts off the ability of the lymph system from the arm connecting to or draining into the rest of the lymph system. So it's possible that post-surgery, people can end up having lifelong problems with swelling, which can be extremely painful in that arm as a result of the damage to the lymph system that was done. So that's a very negative consequence. Dying of cancer is also a very negative consequence. So those are kind of a trade-off that we have in our current practices in medical interventions for cancer. All right, so let's talk about some of the cells that participate in the immune system. Right, all of the cells that make up the immune system are formed from stem cells in the bone marrow. This is great because it means that we can continue making new cells throughout our lives. That's super important. Like I just said, red blood cells have a finite lifespan. We need to keep making more. If you lose a lot of blood, you can make a bunch more and make up for it. A lot of the immune system cells have a very short lifespan. And so we need to be making them on a pretty constant basis and also make a whole bunch more if we are actively fighting an infection. So we group all of the cells that are circulating in the bloodstream as coming from lymphoid stem cells or myeloid stem cells. So bone marrow stem cells differentiate into one of these two kinds, and then the lymphoid stem cells go on to make lymphocytes. Lymphocytes come in three kinds: natural killer cells, B cells, and T cells. They're fairly small cells. The natural killer cells are part of innate immunity. The B and T cells are part of acquired immunity. We'll talk about them more in a few minutes. The myeloid stem cells make everything else. All right, so platelets and erythrocytes are cellular elements that we talked about previously. Leukocytes, lymphocytes versus leukocytes versus erythrocytes are all of the other kinds of white blood cells. White blood cells are going to come in a couple different kinds. There's ones called neutrophils, basophils, eosinophils, and monocytes. Monocytes are going to live in the spleen, and then they're going to differentiate into macrophages. So macrophages are one of the major kinds of phagocytic cells that we're going to find in the body, and then dendritic cells. There's another kind of cell that isn't shown on here because it's not a blood cell. It's called a mast cell. Mast cells and dendritic cells are going to be the kinds that are embedded in surfaces and tissues. Right? So they're not going to end up being circulating in the bloodstream. Neutrophils are the main kind of, the other main kind of phagocytic cell, the vast majority of circulating white blood cells are neutrophils. They have a really short lifespan, only about a day. And they do a few different things as well as doing phagocytosis. They're capable of migrating in the body. So a lot of these immune system cells are capable of moving from circulating in the bloodstream and then moving out of the capillaries and into the tissues wherever they're needed. Right? So they're going to wiggle their way through the capillaries and crawl into places in the body where they're needed. Kind of creepy. They're also going to secrete a variety of different molecules, both chemical messages, cytokines, and also one way that they can cause harm besides consuming bacteria is to secrete molecules that are going to cause harm. One of the kinds of molecules, they're sort of all called toxins, but one example is reactive oxygen species. So remember how when we have oxidative reactions, those oxygen molecules are going to attack other molecules in order to do chemical reactions with them. And that's actually one of the things that these neutrophils do. So when we're out there taking all of our antioxidant nutritional supplements, that's because at some level inside our body, some of this oxidative damage is going on all the time. And basophils and eosinophils are also going to be secreting kinds of molecules that can cause damage or can cause allergic reactions in the body as well. For example, basophils secrete histamines and are often responsible for our experience of allergic reactions. Eosinophils also have a bigger job of attacking multi-celled parasites than other. They sort of snuggle up to them and secrete these toxins, which sort of erodes those larger parasites from the outside. All right, so when we have our immune system cells, the first thing they have to be able to do is recognize a pathogen. We're going to start talking about the ways that our innate immune system can do it. So remember that we're going to have the ability to detect sort of virus and bacteria. All right, so we want to have some kind of chemical signature that many of those kinds of pathogens are going to have. Bacteria or single-celled eukaryotic pathogens are going to have certain characteristic molecules that are part of their external structure that are not found inside the human body, and that these receptors in the immune system cells are going to be able to detect those, right? And when I say detect, I mean they have a matching structure that's going to bind to those characteristic structures. So pathogens have what are called these pathogen-associated molecular patterns, PAMPs. And those are kind of signature molecules that only pathogens are going to have. So certain lipopolysaccharides that make up the cell wall of bacteria. Flagellin is a molecule that flagellates have. So we may have white blood cells that have these receptors that are on their surface that bind to those particular molecules. And they say, oh, hey, some bacteria is here. Hey, some flagellate is here. So these receptors labeled TLRs in this figure are basically proteins sticking up from the surface of the white blood cell that are able to bind like enzyme and substrate to these PAMPs. Right? So these are called pattern recognition receptors. A whole bunch of them that were first discovered in insects are called toll-like receptors, or TLRs. Toll receptors are found in insects first. Then when we found similar ones in vertebrates, they were called toll-like receptors. Real creative people. All right, so there's a whole bunch of different ones. There's genes. Remember, each of these proteins, there's a gene that codes for it. How do we tell we have viruses that have infected the body? Their signature molecules are patterns in their DNA and RNA. And so once those capsules have been endocytosed and disintegrated, then the TLRs are actually inside those lysozymes or lysosomes, and they're going to be able to actually bind to the viral DNA or RNA. All right, and so then a cell response is going to happen to that. All right, so one of our signature vertebrate innate immune responses is called the inflammatory response. So if you think about when you've gotten like a cut that got infected, what happens is it gets red, it gets hot, it swells, and you feel pain, right? And if you're unfortunate, then if it gets really infected, you can get that whitish kind of gook that is oozy, and we call pus left over. All right, so how does this happen? Well, the immune system's trying to fix the problem. And so the redness, heat, swelling, and pain are actually caused by what happens when your immune system responds to the infection. And so you get a cut, a breach in your skin. So this is some splinter, little piece of pointy wood sticking into your skin. It is covered by bacteria because everything is always covered in bacteria. Like little things that look like the sprinkles you put on ice cream, which my dad, the chocolate ones, he always called chocolate bacteria. I want to have some chocolate bacteria on my ice cream Sunday. That sounds great, Dad. And so we have embedded in all of our tissues at our surface all the time, part of the innate immune system, macrophages and these mast cells. So the macrophages are going to, using those receptor proteins, are going to be able to start detecting and engulfing the bacteria, but we want to bring more components of the immune system to help fight the infection. So we want to upregulate the response to this infection. So the macrophage is going to release chemical signals, right? Some of those cytokines. One of the jobs that they do is to attract other immune system cells. So here we have the capillary passing through the nearby tissue. We've got some of these phagocytic cells here, like neutrophils. And what the mast cell is doing over here is it's secreting chemical signals, other kinds of cytokines. It's detecting the presence of the bacteria, and it's going to trigger the inflammation. So it's going to release chemical signals, molecules called histamines. And if you've heard that word before because you have allergies and you take an antihistamine medication, that is why. What the histamines are going to do is cause changes in the capillary. So look at what's happening to the capillary on the left-hand panel compared to the middle panel, right? It's increased in diameter, right? So we have vasodilation, increase in diameter of the blood vessel. That's going to increase blood flow to the area. Increase in blood flow to the area causes the redness and the heat, right? It also makes the blood vessel leaky, right? It causes it to have more of the plasma is going to move out of the capillary. It makes it more permeable, right? So we're going to see these little gaps form in between the cells that make up the capillary. So that's going to cause fluid, plasma to come out of the blood vessel. That's going to cause the swelling. So we're going to get excess interstitial fluid pressing into the tissues in that area. The pain often comes from that swelling. It sort of presses on the nerve receptors. There's also other hormonal messages that are happening. When we have tissue damage, they get released, they send signals to the nervous system. Some of that is going to cause that sensation of pain you get. What else is happening here? All right, so you have a whole bunch of these phagocytic cells that are getting attracted to this region. They are going to eat up all of the bacteria, and then mostly they're going to die, right? So these phagocytic cells, all of the immune system cells don't have a very long lifespan. They are actively lysing or destroying the bacteria. That does cause damage to the cell themselves. And so basically after they've been fighting the infection, they're all going to die. So if you get a huge pileup of these white blood cells at the site of an infection, and they aren't getting rapidly destroyed and reabsorbed by the body, then you get pus, right? This leftover fluidy stuff. It's all the white blood cells that gave their lives to help save you. It's not disgusting. It is a natural product of your immune system at work. All right, okay, so the other thing, mast cells also release a molecule called heparin. Heparin is an anticoagulant and anti-clotting factor, right? So it's going to keep you from getting a blood clot in this area. It wants to keep the blood flowing to this area so it can deliver these phagocytic cells. All right, let's talk a little bit more about these cytokines. So there's dozens of them. Some of the different jobs that they can do, we'll see some more of these later on, that we're going to have response of a cytokine release on another cell is going to cause more cytokines to be released. Interleukins are a class of cytokines that are going to be a way that the immune system cells talk to one another and they say, hey, there's an infection. hey, call your friends, turn everything on, right? So these interleukins are really important in communication between different types of immune system cells so that they upregulate or activate, amplify the immune response. Like we just talked about, one of the functions is that they can attract other immune system cells to the area of the infection if it's specific to a certain area. We're going to see that activation is a word that's important in the acquired immune system. B cells and T cells have to be activated. And that means that other cells in the immune system have to release cytokines that tell them, hey, it's time to go to work. Right now, we don't want to have a big immune response to nothing. We don't want to have the boy who cried wolf, that there's going to be this huge immune response to like one bacteria that's in the body. And so having this set of steps in order to turn on the acquired immune system, some other kind of immune system cell, some of the innate immune system cells have to first detect the pathogen, then draw attention to it, to the B and T cells. And then when they sort of match up and get trained what pathogens to look for, the B and T cells will activate, which means they're going to start dividing, right? And once they divide, and they can turn into different types of cells. So this activation is going to be to trigger cell division or multiplication, right? We're amplifying, just like the pathogen starts to increase in numbers exponentially, the immune system cells are going to start to increase in number exponentially in order to try and fight that infection. Right, mutually assured destruction, escalation. Right, so another example are these histamines that can cause inflammation. The mast cells that are in your nose and sinus, those are the things that when they're bumping into pollen, start to release histamines and cause that sneezy, watery eye, stuffy head problem. Interferon is a molecule that's going to be secreted by cells that have become infected by a pathogen. And it's going to tell neighboring cells, hey, turn your metabolism off because you might have a virus in you. So it interferes with viral replication. It can be used as a treatment in certain kinds of viral diseases. The thing that causes fever in your body is actually a message from cytokines called pyrogens. And they reset the body's thermostat at the hypothalamus. And these cytokines are also responsible for these other kinds of things that we feel, right? So they're interacting with the nervous system as well as the immune system, causing changes to happen in the sensations that we are experiencing in the brain that contribute to all these feelings, pains, and whatever that maybe are not directly caused by the disease, but our immune system's response to tell the nervous system to do something. All right, so lots of communication is happening in the body overall between the immune system and other systems. All right, so let's get down into a little bit more nitty-gritty about what happens to a pathogen such as a bacterium that is inside the body. So we do have the bacteria shown here. It's got these little yellow things sticking up from it. So those are those PAMPs. Those are things that are proteins or other molecules that it's possible to recognize as belonging to a bacteria in general. All right, so we're going to have endocytosis. That puts the bacterium into a vacuole or a little ball of membrane. The lysosome, a lysosome, which is filled with digestive enzymes, is going to be fused with that vacuole, and that's going to cause the bacteria to be digested, right? So it's going to be chopped up into these little fragments or bacteria confetti. Now, these little chopped up fragments are all pieces of the pathogen that could potentially be used in order to recognize the pathogen in the body elsewhere. All right, so once we've chopped up the bacteria, each of these little pieces of bacteria confetti, we're going to call it an antigen, right? An antigen may itself be still a fairly complicated molecule with lots of different subcomponent parts, okay? So each of the antigens, or these little chopped up pieces, may have these other little different specialized regions sticking up on them, and we're going to call those epitopes. All right, so the receptors that the macrophages have may not necessarily be the same receptors that other parts of your immune system are going to use. Right? So we're going to see that the B and T cells are going to have different receptors that can and do match up to these antigens. All right. So what the macrophage is going to do is inside the cell, it has these other molecules called MHC2s. All right, that stands for major histocompatibility complex. The kind is class 2. There's also class 1s. We'll see them in a minute. So they stick these antigens onto these MH2s and they move them to the cell surface. All right, so this is called presenting the antigens. Basically, it says I chop this pathogen up into confetti and here's a piece of it. All right, and it's sticking up on the surface. Why does it want to stick it up on the surface? Because the B and the T cells are going to be cruising around looking for these presented antigens, right? So these presented antigens are the way that the innate immune system has of telling the acquired immune system that there is a particular pathogen in the body and hey, you need to respond to it. All right, so these macrophages and other types of innate immune system cells are going to do something called presenting the antigens, right? So once they have detected a pathogen, they're going to be considered an antigen presenting cell, or APC, right? So we're gonna start to look at, okay, how do these B and T cells recognize these epitopes? How do they do it? What do they do? And also when we talk about vaccines, the vaccines are going to also be things that recognize these antigens, and again, more specifically, the epitopes. All right, so these MHC class 1 and MHC class 2 molecules are important. We looked at this example of macrophages that are going to be presenting antigens using MHC class 2 molecules, right? And antigens that are presented by monocytes, macrophages, dendritic cells, or B cells, cells of the immune system, right? So antigen presenting cells are going to be cells of the immune system. And what they're going to be holding up in those MHC class 2 molecules is something that they have decided is non-self. All right, so these MHC class 2 antigen presenting cells are, the purpose is to recognize things that do not belong to our own bodies, right, that need to be attacked. There's a slightly different version of these antigen presenting structures, molecules, called MHC class 1. MHC class 1 molecules are used by cells that are actually cells or platelets. Every normal cell of the body that's, and even these immune system cells. Right, so this is a cute little epithelial cell here. Platelets also have them. Erythrocytes don't. Erythrocytes don't have a nucleus. They can't make them and produce them. Right, so these MHC class 1 molecules are basically, if we have an antigen presented on an MHC class 1 molecule, it's a cell of our own body saying, I'm being attacked by this thing. Okay, so we have different types of cells in the body that have different jobs in terms of saying either I'm a cell of your own body, hey, but I'm being attacked by this thing and here it is, versus I am a cell of the immune system and I have discovered this thing that is not a self and it needs to be attacked, right? And again, MHC stands for major histocompatibility complex. We have other histocompatibility complex molecules in our bodies. So there's a whole bunches of different ones, and red blood cells have them too, and they are responsible for the concept of blood type, right? If we get a blood donation from somebody, it has to be the same type as us, otherwise the blood coagulates. It's an immune system response. All right, so anytime somebody needs to have any kind of organ transplant, the organ transplant has to come from a donor that matches histocompatibility markers. You're compatible, right? So similar genetically related individuals are more likely to have the same immune system markers. Blood types are there's not that many of them, but there's a whole bunch of other kinds of histocompatibility complexes. So if you have volunteered as a potential match for organ donation or for bone marrow donation, your blood will be screened for a whole long list, not just blood type, but a whole bunch of these other factors. And the most factors that match will be the best match for the donor, or so the recipient. Typically, they still have to take immunosuppressing drugs for the rest of their life anyway. All right. All right, our last example of innate immunity is, what do these natural killer cells do? Well, they kill cells. They're natural born cell killing cells. Right, and how do they do that? How do they know which ones to kill? Well, so your healthy cells, normal cells, are going to have a certain pattern and number of MHC1 molecules. And so the natural killer cell is going to be circulating around in the bloodstream, bumping into other cells. It bumps into a cell and binds to it that has normal pattern of MHC1. It's going to let it go and say, hey, have a good day. If it bumps into a cell that has an abnormal number or pattern of MHC1 molecules, right, then it's going to kill it, right? So it's going to be able to detect a variety of different kinds of cells that are infected with pathogens. And cancer cells typically have abnormal MHC1 molecules. And so these natural killer cells actually are going to destroy cancer cells, hopefully before they grow in numbers enough to become a tumor, especially to spread to other body organs. And in fact, your immune system kills cancer cells in your body an average of 1 a day. All right, so it's really only a matter of time before any of us is going to actually have some cancer cell somewhere in our body that isn't detected in time and is able to grow into a tumor that's a problem. All right, so the way the natural killer cells do this is that they release that perforin and granzyme, and it's going to cause destruction of either the infected cell or of the cancerous cell. All right, our last major topic we're going to talk about is acquired immunity. So shifting gears from innate immunity to acquired immunity. Acquired immunity is going to happen only using the cell types, the lymphocytes, B and T cells. And not only is it going to be specific, but it's going to attack pathogens right now, but it's going to create a stored memory to prevent future infections. B cells are responsible for what is called the humoral response. That means it's circulating in the bloodstream. The way it actually fights the pathogen is by releasing antibodies. Antibodies are either going to neutralize, destroy, or target for destruction the pathogen, and it's going to create B memory cells that are going to keep the ability to multiply in the future and release antibodies rapidly if there's a new infection. T cells are called the cell-mediated response because they attack pathogens cell by cell, but the way they do it is to attack your own body cells that are infected by that pathogen. There are actually 3 kinds of T cells, and so not only are they responsible for killing infected cells, but they're also responsible for amplification and retreat, or suppression of the overall immune system. They're very active with releasing cytokines of different kinds. And so these helper T cells, right? So the 3 kinds of T cells are cytotoxic T cells, abbreviated capital T sub C, helper T cells, abbreviated capital T sub H. Their job is going to be to activate the killer T cells and the B cells. We talked about that earlier. What is activation? It means that the killer T cells and the B cells will be stimulated to divide and differentiate. Suppressor T cells, we aren't really going to talk about too much, except you need to know that they exist, capital T sub S. And what happens is that they're able to detect when the pathogen is decreased to a safe level in the body, and they're going to release cytokines that instead of amplifying the response, they shut down the response, right? Now remember that most immune system cells have a very short lifespan in the body. And so if we aren't actively turning more and more of them on, they will fairly rapidly decrease in numbers just by dying. One trademark of having an infection or being sick is if you go to the doctor's office and you get a blood sample taken, you'll have an increased level of white blood cells circulating in your bloodstream compared to what a person has when they're healthy. T cells are a little scary because if they're running around in your body attacking things that are actually part of your own body, then they're going to you'll have an autoimmune problem. So what happens to the T cells is that actually when they are released from the bone marrow, they're going to go to the thymus, which is why they're called T cells. And the thymus has an ability to, if the T cell is binding to or sticking to cells of your own body, that they'll destroy it. So those T cells, hopefully, that are self-reactive, or that are bad in that they're going to be wrong in deciding what is self versus non-self, that hopefully those are destroyed. So there's an editing process. All right, next couple slides talk about a really amazing system that allows the B and T cells to recognize any pathogen that exists that the immune system has never seen before, right? How the heck can it do that? All right, this is a really amazing system. If your job was to create a system that could do this, I am not sure that anybody would have come up with this. It's really neat. All right, so just like the phagocytic cells, they have receptors. They look a little different. The B cells have these Y-shaped receptors. There's a part of it embedded in the cell membrane and then a part of it that sticks up. T cells have these like rod-shaped ones, but what's similar is there's this constant region labeled C that's always the same. It's not the part that interacts with the antigen. And then there's the variable region. The variable region is at the tip, and it's where the antigen is going to bind. Right? So remember that the antigen and the receptor have a lock and key fit, and so they have to match to one another. So how can a cell produce a receptor that could possibly bind to some pathogen it's never seen before, and it doesn't know how it's actually made? Well, it just makes a whole bunch of different ones at random, and it hopes that one of them will match to that future pathogen, right? So what's really neat about the way that the antigen binding sites are made is that it has a system that generates random variation in the structure of these proteins, which makes them have a different shape. Okay. All right. So I'm going to give you one example of how that works. And one thing to keep in mind is that within each B cell, when it differentiates, there's editing of the DNA so that each B cell has a gene that makes the variable regions, or two genes, one on each side, two different ones. And that every receptor that's on the surface of a single B cell will have the same of these two genes. So once it's differentiated, each B cell is only going to make one version of the receptor. Right? Same thing for the T cells. So all these little receptors on the surface of any one B cell will all be the same. But if we look at all the different B cells in the body, there's literally millions of different variations of what these antigen binding sites are going to look like. Right? So we've got this population of cells that are pre-programmed or built so that they could recognize any one of a huge variety of different potential antigens. So the way that happens is through splicing of the DNA. This is one example of the inner side of the variable region for B cells, and it's called VDJ recombination. So this is kind of bio 230 stuff, cell molecular stuff, but I need to cover it at a very big picture level so that you understand at least a tiny bit of how this process works. So the original complete genome of these cells has 50 different little sections in the genome that are these V segments. It has 27 different genes, basically, or sections of the genome that are the D part and 6 that are the J part. As the DNA is being processed during cell differentiation at random, I have no idea how that actually happens. That's bio 230 stuff. At random, one of these V segments ends up getting left, not cut out, but left behind. One of the D segments ends up getting randomly left behind, not cut out, and one of the J segments. All right, so out of all these 50 plus 27 plus 6 different sections, there's over 8,000 combinations just to make this inside part, right? And so the receptor actually is made of these two parts, the outside and the inside. So if you take all the possible combinations of the outside part and match them up with all the possible combinations of the outside part, you get over 10 to the 6th different combinations. It's a big number, right? So we have this random reassembly, and that causes this tremendous variety. So when we think about human populations, and we think, okay, each individual has the possibility of detecting all these different versions. Each of these V segments, D segments, and J segments is possible, has mutations in it so that they differ slightly from one another. Right? So each individual person may have slightly different versions of these genes that, that code for these proteins. So within a population, you may have the possibility of creating different antigen receptors for the B and T cells because of mutation that's occurred over human history. So we see different people with different abilities to generate these receptors that might be better at binding to certain diseases versus other ones, right? So another example, a little more detailed idea of why genetic variation in a population in the immune system is beneficial for the population and for its evolution as a whole. whatever disease or pandemic hits us, hopefully there's somebody who's got the genetic ability to make receptors that bind to that disease and it doesn't kill everyone. All right, so what happens when an antigen receptor binds from one of our B cells or T cells? So this example is showing what happens to B cells. Same thing happens to T cells. All right, so we're showing these B cells that differ in antigen specificity, right? That means, and it's showing these receptors as looking really different from one another, but it's really just that the little fork at the tip. All right, so we've got some antigen molecules, right? We've got some confetti, bacteria confetti that the macrophages, right? So they also like spew some of it out into their surroundings. And so these B cells could bind to it separately as well. And we have this particular antigen, this B cell in the middle with the purple receptors binds to it, right? Antigen receptors on these other two B cells are different because of that recombination process. They don't bind to it. So these B cells, if they get activated, are going to divide, right? And this process of only the cells that respond to the antigen, a specific antigen dividing, is called clonal selection. So it's kind of, if you think about how natural selection works, natural selection is increasing, right? These cells, these particular B cells that bound to the antigen, right? So we want to increase in number the ones that are capable of recognizing this particular pathogen. Same process of clonal selection happens for helper T cells and cytotoxic T cells. So just because I'm showing you this B cell slide doesn't mean it isn't also happening, right? So all the B cells and T cells circulating in your bloodstream, innate immune system finds, chops up pathogen. So the antigens are floating around. At least one B cell, one helper T cell, and one cytotoxic T cell have to bind to it in order for the entire acquired immune system to turn on. Once one cell of each does, there's a whole cascade of things that happen. So those cells are going to start dividing like crazy, so that there's lots of them circulating in the bloodstream, moving into the tissues, that there's enough of them that they can find and kill all of the pathogen. During the process of clonal selection, as the cells are dividing, some of them turn into or differentiate into memory cells. Memory cells don't do anything right now, they just hang around. And they can live for years to tens of years to, in some cases, the rest of your life. Not always, not necessarily. And then the other ones, though, that are going to help fight the current infection are going to generate a whole bunch of these receptors and then just let them loose. Right, so they're basically taking those receptors, releasing them into your circulating bloodstream, and now we call them antibody molecules. Okay, so they're basically free-floating versions of those receptors. And we'll see what they do in a couple minutes. So looking at what happens with the T cells, helper T cells actually have to bind first. So the helper T cells, what they're going to bind to is an antigen-presenting cell. They're going to bind to an antigen-presenting cell that is holding up an antigen, presenting an antigen on a class 2 type MHC molecule, right? So that means the helper T cells are going to bind to one of these other kinds of immune system cells. And helper T cells have a protein called a CD4 that is sort of how we define them as a helper T cell. And it's going to bind to the receptor of that particular helper T cell. If it matches and binds to the antigen that the other cell is presenting, right? It has to be able to match to it. It's going to create a firestorm of cytokines being released, right? So the the purple cell here, the macrophage, is releasing these purple cytokines. It's going to attract these helper T cells. If the helper T cell successfully binds to the antigen that's being presented, it's going to secrete a bunch of cytokines, right? These cytokines have to do with regulation. It's going to cause a positive feedback loop. That means it's going to activate the helper T cell itself. So it's kind of like autocrine signaling. And then it's also going to activate B cells and cytotoxic T cells. And so the helper T cells are not just helpers, they're actually critical to the whole process getting turned on. B cells and cytotoxic T cells have to be activated by a helper T cell before they start dividing and differentiating. So this is a little bit of a quality control step. We don't want to, if there's just one cell somewhere in the immune system that has found this antigen, we don't want to turn the whole thing on, right? So it's a little bit of a step that's going to prevent sort of overreaction of the immune system. All right, so remember that we have class 2 MHC molecules and we have helper T cells that have a thing called a CD4 protein that's going to bind to those class 2 MHC antigen presenting cells. Our cytotoxic T cells, we kind of define those as ones that have a CD8 protein, and they're going to be able to bind to class 1 MHC molecules. So the cytotoxic T cells, what they're going to do is they're going to bind to a body cell that is infected with the pathogen, and it is presenting this antigen. Remember, the class 1 MHC is saying, hey, I am infected with this thing. Okay, I need to be killed. And that's what the cytotoxic T cells do. They come, they bind to it, they secrete granzyme and perforin, just like the natural killer cells do. That's going to cause pores, and it's going to lyse or kill the infected cell, right? CD8 T cell can possibly go and do this again. So we're going to kill cells that are infected with the pathogen in the hopes that the pathogen then it's not able to reproduce, and that we don't spread the infection to other cells, right? But if your cells, or a lot of your cells, are infected and your killer T cells are going around killing them, your body's got a lot of work to do. It's got to replace all of those dead cells. So that's another reason we feel tired when we're sick. All right, so when we look at what those B cells are doing, remember they release antigen, or they release antibodies. Antibodies, how they help fight the pathogen is a little bit different depending on what kind of pathogen it is. Viruses are really small, and that means that when the antibody binds to them, it sort of covers them all over. And the way the virus has to function is to bind to a host cell. So if it's covered all over with these antibodies, it can't bind to the host cell. And so this is called neutralization. It isn't actually killing the viruses, but it's preventing them from being able to attack your cells, right? So other small molecules, relatively small molecules that may be released by pathogens, which we'll just call toxins. So some of the things that make you feel sick in certain types of infections. Antibodies can also bind to those toxins, and if the toxin is the thing that causes harm to your body, then it removes it, it prevents it from causing harm. All right, so slightly larger things, pathogens, bacteria, the antibodies are going to bind, but what they do is that they're going to target the bacteria for disposal by the macrophage, right? So suppose this bacterium just doesn't happen to have any of those general markers on its surface that the macrophage can detect with those TLRs. The antibodies may be able to detect it because they're looking at these specific antigens that are that have been presented, or they have a more sort of detail-oriented way of looking at the pathogen. So they're going to bind to those antigens, and then anything that these antibodies are bound to, the macrophages also are able to recognize and consume. Right? So that's called opsonization. It's really a way of just marking for marking for destruction. Right? You're going through your garage and you're putting a big X on boxes that you want to throw everything away in them. Same thing, same idea. All right, the other thing that antibodies do, and these are for eukaryotic cells. So eukaryotic cells that see where the antibodies are, they are binding to the surface. What they're going to do is attract those proteins from the complement system. Right? So those complement system proteins are floating around in the bloodstream all the time. If they bump into the way these antibodies are sort of arranged on the surface of the foreign cell, they're going to stick together. Some chemical transformations are going to happen, and they're going to cause this set of steps that lead to formation of the attack complex. Right, so it's going to punch a hole in the cell membrane. This is how our immune system can attack the actual foreign cells. Right, so remember, cytotoxic T cells, they can't bind to these foreign cells. They only bind to self cells. So the formation of this complement system is the way that we kill eukaryotic parasites. Antibodies are a focus of immune system study for a variety of reasons. One is they're circulating in the bloodstream. So one way we can measure whether somebody has had a certain infection is to detect antibodies to it in their bloodstream. After you've been vaccinated for a disease, your body mounts an immune system response that includes producing antibodies. That's the purpose of the vaccine. So we should see antibodies circulating in the bloodstream after somebody's been vaccinated. There's different kinds of antibody molecules produced by different cells that have slightly different functions. And they're also called immunoglobulins. So remember, they're proteins, and they're called things like IgG, IgA. There's different ones. The Gs are the most common ones that we find after a strong immune response has been mounted. The As are ones that are produced earlier in your immune response. They may not bind to the pathogen very well. So when we talk about how well a vaccine works, it's going to depend on how strong immune response is mounted by your body, how many antibodies are produced, how strongly those antibodies bind to the actual pathogen, and how long that immune response stays in your body after you've had the vaccine. Also, these antibodies can be transferred to a developing fetus through the placenta, and they can also be transferred to a baby from the mother via breast milk. So we have the ability of a mom to pass on some of her immune system's memory and also current immunity to her baby. All right, so when we think about how these memory cells work, right, so not only B cells, but helper T and killer T cells or cytotoxic T cells make memory cells. So when we have our first exposure to a particular antigen or a disease, look at the days along the bottom here. This is how long it's been since we've been exposed to that infection. And we have a little delay, about a week to 2 weeks before we have the highest, we start producing antibodies, and then we have a high number of antibodies to that particular disease. And then as we fight off that infection, the number of pathogens decreases in the body, we see a decline in the number of antibodies that are circulating. They get filtered out from our by our kidneys, and we break them down. All right, so antibody concentration is on the y-axis here. Now, suppose we get exposed to that same pathogen again, right? Now we have not the antibodies circulating in the bloodstream, but we have those plasma memory cells. So instead of having one B cell somewhere in the body that matches to this pathogen, we have thousands, right? So when they get activated, we go from 0 to 10 to the 4th really fast, right? So notice that we have not only a much bigger response, we have a higher antibody concentration, we also have it faster, right? Less than a week. We have a way higher level. So when we have a second infection by the same pathogen, we actually mount an immune response to it that's so fast and powerful that we can hopefully kill all the pathogen before they increase in population numbers enough to make us sick. Right? So when we have a second exposure to the same disease, we may not get sick at all. However, that response is specific. If we have an exposure to a different disease, we have a pattern that looks the same as we had to this disease A in the first place, right? So it doesn't give us protection from a different disease. Now, diseases that are very similar to one another, sometimes we'll have cross-reactivity. That is, if we've had dengue fever, then we get some protection from yellow fever or vice versa, because they're similar enough to one another that antibodies for one of the diseases will be able to recognize, maybe not as well, but somewhat, right? They won't bind as strongly. And so they'll be able to help protect us from a similar disease. All right, so we have this process, and that's how vaccines work, right? So what a vaccination is going to do is it's going to expose you to the disease, hopefully in a way that doesn't make you sick. And it's going to do this little initial response. So your body is going to make memory cells. And so when you are exposed to the actual live disease, you're infected with it from somebody else who's sick, you have this response right away, right? Instead of having to get sick and then wait for it, right? So the idea, the principle of vaccination is to give you this little initial exposure so your body mounts a large immune response to it, so that when you are actually exposed to it, then you are protected from it already. The way that this is done is to expose the person to the disease in some way that hopefully doesn't make you sick, right? So if you think about how the immune system works, we have to somehow introduce an antigen, the antigen for it, right, without the organism itself. Right? So vaccines are formulated in such a way that the antigen, or sometimes the toxin that's produced by the organism, is introduced into the body, a small amount of it, not enough to make you sick, not in a way that's the actual disease, that will cause the immune system to have a response to it. Right? So sometimes we feel a little sick when we've had a vaccination. That's your immune system responding to it. Right? So that's actually good. All right. So we'll talk a little bit more about different kinds of vaccines and how different populations might respond to them, or the genetic diversity in immunity in our live session. And we're just going to sum up with an overview of this whole system of acquired immunity. And it's going to begin with a disease-causing organism coming into the body. So somewhere in the body, there will be this antigen is present. Typically, it's not the whole organism. It's going to be sort of chopped up due to the work of the innate immune system. At least one B cell, at least one helper T cell, and at least one cytotoxic T cell has to be able to bind to that particular antigen with those randomly created various antigen receptor proteins. Right. So once the helper T cell has bound to that antigen, it's going to become activated. It's going to start dividing, and it's going to start releasing these cytokines, right? Those cytokines are going to activate the B cells. Cytokines are going to activate the cytotoxic T cells. The cytotoxic T cells are going to divide. They're going to do the clonal selection process. Those cytotoxic T cells are going to directly attack the cells of your own body that have been affected, so the antigen presenting cells. It's also going to make memory cells, and those will stick around. Helper T cell is also going to make memory cells. B cell is going to start to divide. It's going to make memory cells. It's also going to make those plasma cells that are going to release the antibodies, and the antibodies are going to help either neutralize or target bacteria for destruction, or the antibodies are going to destroy the pathogen. All right, so we have these different things that all have to happen. It seems like it's unlikely that it could possibly happen, but it does. That we have this incredibly flexible, responsive immune system. There's a lot of overlap. There's a lot of redundancy. And it's really amazing and quite complicated. So a lot of the research that's been done on how the immune system works at a detailed level has actually been done since the 1980s. So when HIV and AIDS came into the human populations and became a large source of mortality in the United States and other developed countries, because it's the way it is as a pathogen is pretty complicated. It really sparked a lot of research into way more detail about how the immune system works. All right, so we do have the possibilities of the immune system going wrong. We have allergies, we have inappropriate immune responses to things that aren't pathogens. And then we also have autoimmune disorders, right? And those are things where, because of a genetic trait or because something that happens during our lifetimes, our own immune system starts to attack particular types of cells in our bodies, right? So we have this great and amazing system that's kind of like can be a loose cannon and cause a lot of harm. But it also ends up protecting us not only from pathogens, but also from cancer, our own cells gone wrong, and that our long lifespans as vertebrates, as mammals, as primates is in large part due to the success of the vertebrate immune system. So I hope you're really excited and interested in how the immune system works, and we'll talk about it more in our in-class meeting.